Hi dear students, welcome to one more session of genetics, and today we are going to talk about a topic called DNA fingerprinting. What is DNA fingerprinting? If you have a look at this particular picture, even if you don't know what is fingerprinting, you can see that when you analyze it, you can identify that there are three suspects and there is a crime scene, and based upon the band pattern of the obtained from the crime scene as well as from these suspects, you can say that the suspect too was being present at the crime scene. But what are these band patterns and why are they significant over here? These are in fact illustrations of DNA fingerprinting, and the DNA fingerprinting technique here is being used for solving a crime mystery. But what are these bands over here? These bands mainly correspond to the DNA which was being obtained from a crime scene, as well as the suspects of the which are being considered of the crime. So, but how do you get these bands of DNA? Of course, we know that DNA as such is when you take a whole genomic DNA and you do a agarose gel electrophoresis, it gives you a single band. But here you have multiple bands, and it indicates that DNA has been spliced by some method or it has been cut into small fragments. And you can also see that in the crime scene you get a DNA pattern, okay, and in the suspect also you are able to find a specific pattern. So, I need to tell you that DNA fingerprinting is a technique, in fact, which enables us to identify a person using his DNA. So, from the term itself or from the picture itself, we can understand that we are not sequencing or identifying the whole sequence here, but you are doing something else. You are cutting the DNA and you are you are running a gel. Of course, that is also there, but some more steps are being present, and these steps enable you to identify a person from the other. So a fingerprint is something unique to a person, especially a DNA fingerprint also. And when it is being coded by your DNA, it is referred to as a DNA fingerprint. Now, what is DNA? We all know it has been present in the genetic material as a genetic material in the nucleus, and within the nucleus, every human has got different chromosomes, and the genetic material mainly constituted by DNA is being present in the chromosomes, especially inside the nucleus. Now, why do we have to take DNA, and why does it constitute uniqueness when it go to consider every individual, or why do we get a fingerprint from DNA? That's what we are going to explore at the beginning before then, rather than rushing into the topic. If you go to consider every DNA of each and every human individual, you can see that 99% of the DNA is found to be similar, because most of the DNA codes for essential functions, and almost all of them, almost 99% of the DNA in the human beings has been found to be similar, and it is only the remaining 0.1% is being found to be different. And unless and until you are an identical twin, this 0.1% is the difference that you have. So, although this will sound like a small amount, it means that there are around 3 million base pairs that has been different between two people. And using these differences, if you compare these differences, it will help us to distinguish one from another person. And why does this difference come? this difference, it mainly comes because of the presence of certain repetitive units or repetitive DNA sequences in the genomic DNA. And so by comparing this repetitive units or these variations and correlating them with techniques such as DNA fingerprinting, we can identify a person from the other. Now, let me just talk a few words on what are repetitive DNA. If you go to consider repetitive DNA, they could be tandem repeats, terminal repeats, as well as interspersed repeats. And tandem repeats, they are highly repeated sequences which lie adjacent to each other without any interruption. And if you go to consider the tandem repeats in the vertebrate genome, they could be mainly of like you can have satellite DNA, and also you can also have a repetitive DNA. I think it's a bit confusing. You can just have a look over here. If you go to consider the repetitive elements of the whole genome, okay, they could be of those ones. There are many of them, but I'm just concentrating on the ones which are tandemly repeating. Okay, the most important one among them are the tandem repeats as well as the satellite DNA. Now, what are tandem repeat DNA? Tandem repeat DNA is a stretch of DNA which is not being interrupted with each other. Now, if you have a sequence called GACCT, you consider it's just a example I am saying. GACCT, multiple copies of GACCT will be repeated and they will not be interrupted by another sequence. That is a tandem repeat. Sometimes the repeats might be disturbed by other sequences. That is referred to as an interspersed repeats. And if the repeats have been found towards the ends of a chromosome, it is referred to as the terminal repeats. So, when you go to consider the tandem repeats or the repetitive DNA in general, these repetitive elements can be mainly divided into two called the repetitive DNA as well as the satellite DNA. And these satellite DNA as well as repetitive DNA have some major differences. If you go to consider the repetitive DNA, these are they mainly refers to the stretches of DNA that will repeat throughout the genome, whereas satellite DNA refers to some short repetitive DNA sequences mainly being found in the centromeric region of the genome. And the repetitive DNA can be of two types, either tandem repeat or interspersed repeats. I told you earlier what is the difference between a tandem repeat and an interspersed repeat. And a tandem repeat is a stretch of repeats which is not being interrupted by any other DNA sequence. That is, if you have a sequence like GAATTC like that, that repeat is multiple copies of that is being placed and no other DNA sequence comes in between. That is a tandem repeat. But in an interspersed repeat, you have something interrupting it. You will have an illustration in the coming session. So in the case of satellite DNA, they are tandem repeats, and in the case of repetitive DNA, it is mainly consists of few base pairs to hundreds of base pairs in the repetitive unit, but the satellite DNA is a little bit shorter and it will have only up to 60 base pairs long. And in the case of repetitive DNA, they are moderately repetitive with about 10 to 105 repeats per genome, but in the case of satellite DNA, always these segments, though they are being small, almost greater than 106 repeats per genome will be found in the satellite DNA. And repetitive DNA is mainly being located in the intergenic regions of the genome, and satellite DNA is located on the specific chromosomes as well as the centromeres of the chromosome. And two main types of repetitive DNA are the tandem repeats as well as the interspersed repeats, as we had told earlier. And among the satellite DNA, you have mainly two types of again classifications called the microsatellites as well as the mini satellites. And A repetitive DNA, it serves as an evolutionary device analyzing the formation of new genes, whereas satellite DNA play a major role in fingerprinting. Repetitive DNA appears as light bands, but satellite DNA gives small dark bands. Now, we had started from this picture where we are trying to find out the variations between different DNA strands of different people. And we have come across to a region where we are talking about the repetitive DNA and the satellite DNA. We should keep in your mind for the process of DNA fingerprinting to happen, basically within the DNA, there do exist certain differences. And these differences are mainly being constituted by these repetitive elements. And the repetitive elements mainly consist of the main ones. There are different types, but most important among them is about repetitive DNA and the satellite DNA. And just now, we just explained what is the difference between repetitive DNA and satellite DNA. And it is the variations in this satellite DNA, specifically talking, which enables us to yield a process called DNA fingerprinting. So, that is a case of that is the significance why I'm talking about the repetitive DNA as well as the tandem repeats as well as the satellite DNA here. And among the satellite DNA, you have what? Two main types, like you have microsatellite and mini satellites. We'll be going into that. So if you go to consider the repetitive DNA, or they are stretches of DNA that repeat throughout the genome, and they are mainly being found in the non-coding regions of the DNA present throughout the euchromatin. And this repetitive DNA, as I told you, it could be of two types. They could be of tandem repeats or they could be of interspersed repeats. Now, this is an example of tandem repeat. Now, here a sequence called GATA, that is the one which is getting repeated. That is being now. There are multiple copies. If you're considering three different genomes, you can see that there will be 8 repeats now in the GATA is been repeated 8 times in the first genome and the second genome. And in the third genome, this GATA is been repeated 9 times. So this difference, okay, the number of repeats or these tandem repeats could contribute towards the what? The difference which is being used in DNA fingerprinting of a person. And if you go to consider satellite DNA contains tandem repeats. And just if you go to consider the repetitive DNA as such, what happens? Sometimes it might be tandem repeats. At the same time, they are being found to be a little more longer, but the number of frequency by which they get repeated is being found to be less in the case of repetitive DNA. Okay, so that's what you have. You can think that as they are only a few base pair lengths. Okay, that is, it could be around 100 to 100 base pairs, but this is only satellite DNA is only 60 base pair long. But the number of repetitions which have been present in the whole genome of repetitive DNA is 10 to 105 repeats, but at that of satellite DNA, it has been found to be more. Now, coming to tandem repeats, this is what a tandem repeat is. And after that, okay, let's talk about the mini satellites and the micro satellites. Coming to the satellite DNA, if you go to consider two different types of satellite DNA, mainly you have micro satellites and mini satellites. The main difference between the microsatellite and the mini satellite is based upon the length of the base pairs. In the case of mini base satellites, you have 10 to 60 base pair in the repeating units, and they are mainly being located in the centromeric region, whereas microsatellites, they have less than 10 base pairs in the repeating unit, and they are mainly being found in the telomeric regions. So it is the difference between all the variations which you found in this micro satellites as well as the mini satellites, which will be contributing towards the what? Mainly the fingerprinting technique. It's mainly the differences which contribute, generally speaking, the differences mainly in this satellitic DNA is which is mainly contributing to the fingerprinting technique. And you can just have another illustration over here of the different mini satellites which we are using here. And if you're going to see the mini satellites, now here in the first one, it has been repeated 1, 2, 3, 4, 5, 6 times over here in this individual, whereas in the second individual it has been repeated more times. So based upon this difference at which these sequences are being repeated, variations will arise, and that's is being utilized, or that forms the base or the cause of different bands when we are doing a DNA fingerprinting technique. This is just an introduction on what forms the basis for the differences in DNA fingerprint found in fingerprinting. We will be dealing with the steps as well as the steps of DNA fingerprinting in the next sessions. Thank you for now.